So my name's Rebecca, and I'm going to be talking about some of the genomics work that we've done with the pathogens in the Healthy Trees, Healthy Future project. And this is really about looking at management of these tree diseases into the longer term. So at the start of the program, we started by sequencing the full genomes of around 7 Phytophthora species and a couple of strains of each of those. And the reason for doing this is that we knew that genomics would help us in several ways. So it can help us to understand the genetic diversity of the pathogen populations. It can help us understand the origins of new diseases and maybe even the pathways of introduction. It can provide clear taxonomic information which really underpins our biosecurity system. It can help us with screening for host resistance, as Natalie's just described. It can also help us in the lab when we want to pick our strains for the ones that we want to use to do these studies. And it can also help us in developing diagnostic methods. So if we look at the population genomics of a couple of Phytophthora forest pathogens. So firstly, I want to talk about Phytophthora pluvialis, which is the image on the left. So Lindsay's already described how we know that Phytophthora pluvialis originated from Oregon. So when we sequence the genomes of Phytophthora pluvialis, we sequence one from Oregon and we sequence one from New Zealand, and that allowed us to compare the genetic sequences of those pathogens and find differences. So we found around 27 differences that could then be used to characterise populations of these pathogens in New Zealand and in Oregon. So what we found was that we could identify multiple genotypes from Oregon, which are the ones in blue, or the USA, which were the ones in blue, and isolates, different genotypes of the isolates from New Zealand. So you can see from figure A that we have greater genetic diversity in populations of Phytophthora pluvialis in the USA than we do in New Zealand. But most interestingly, you can see that we have 2 different populations of Phytophthora pluvialis in New Zealand, which suggested to us that we actually had 2 introductions of this pathogen to New Zealand. This allows us to begin to speculate on what those potential pathways of introduction might have been and how we can possibly prevent further introductions of this pathogen into New Zealand in the future. On the right, we're looking at Phytophthora canoviae, and Lindsay has already mentioned how physiological needle blight we can now attribute to a Phytophthora, and this is that species. So again, we've looked at the genome sequences of isolates of Phytophthora canoviae from New Zealand, the UK, and in Chile. In New Zealand, Phytophthora canoviae does cause disease on pine, but it actually causes a lot more damage in the UK on beech and ornamental species, even mortality. And it's also recently been described on some species in Chile. What we found from this analysis was that Phytophthora canoviae actually has a far greater genetic diversity in New Zealand than we see in populations in the UK or Chile, which suggests that New Zealand might actually be the origin for Phytophthora canoviae and maybe even endemic to New Zealand. So this information is really important for our biosecurity. We don't want new isolates of these pathogens here in New Zealand because that could lead to far greater disease. So we've already talked a little bit about Phytophthora cactorum. This is one of the oldest Phytophtheras, one of the ones that was first described in the 1800s. It was found on cacti. But we know that it's been associated with pine in New Zealand for a really long time as well. It's been associated with shelter belts. It's found in nurseries. Now, when we started this research program, we realised that actually Phytophthora cactorum that we see on pine and Phytophthora cactorum that we see on apples, they actually are quite different. They behave differently in the lab, and there's a few differences in some of their genetic sequences. So this actually led to a little bit of work that eventuated in us describing Phytophthora cactorum from pine as a new species, and it's been renamed as Phytophthora aleatoria. And in this bottom tree, this phylogenetic tree at the bottom here, you can see how cactorum and aleatoria genetically are quite different. And this led us to sequencing genomes of Phytophthora cactorum from apple and pine, as well as from other tree species from different parts of the world, and doing whole genome analyses to compare those isolates. So isolates if looking at this figure here, in the yellow, those numbers there are those are the isolates that are most similar when we compare them pairwise. If you look at the red, those are the ones that are quite different. Now, Phytophthora cactorum from pine, or Phytophthora aleatoria as it's now called, is along that bottom row, and you can see that actually it's quite different to most other species. But we're also seeing a really complex picture in terms of their genetics between all of the different isolates, which suggests that this is actually a complex that really needs teasing apart. So this genomics has provided us with some really important clues here, but actually we really need to highlight how important it is to be able to identify species accurately. It's really important for biosecurity. We need to be able to identify the plant pathogens quickly and as accurately as possible. So Natalie's already talked about some of the screening that we've done for looking at resistance and susceptibility in pine. And the great thing about some of these experiments was that we performed these over a time series, so on different days we collected samples and they got put away in the freezer. And then once we had a bit of an idea from those pathology experiments which ones were showing some potential for resistance and which ones were looking a little bit more susceptible to those diseases, we could go back to those samples in the freezer and look at those to understand a bit about what's happening in terms of gene expression in those pathogens, and also looking at those samples for metabolomic analysis. So if we think about the gene expression for those particular samples, what we had was a mixture of the pine DNA and the Phytophthora DNA all mixed up together in a big data set. So we had to pull the Phytophthora DNA out of that data set away from the pine DNA, and that was all done bioinformatically using some pretty cool computer programs. And what we see when we compare the Phytophthora pluvialis and the Phytophthora canoviae data sets is that we have a far greater number of DNA sequences from the Phytophthora when we look at pluvialis compared to when we look at canoviae, which indicates that pluvialis is probably a better pathogen or a more aggressive pathogen on pine than what we see with Phytophthora canoviae. Now, effectors are proteins that help our pathogen, or that enable our pathogens to infect its host. So they're the proteins that actually go in and cause the attack. Now, when we look at the genes that encode these proteins, we also see a similar pattern in that the genes that are present in Phytophthora pluvialis are expressed to a greater level than what we see in Phytophthora canoviae. So this gives us a bit of an idea around the differences between infection between those two species. Now, we haven't really got as far as we would have liked to during the program with this particular part of our research, but one of the things that's been really great is that it's enabled us to strike up really cool collaborations with people at Massey University, University of Canterbury, who are now going to take these genes or proteins from these pathogens and use those with plants like tobacco, which is the lab rat of plant sciences, and to use those in high throughput screening for resistance to disease. So this is where we're looking into the future for long-term solutions to disease. Can we find durable resistance using these kinds of methods? And this has been done with potato, it's been done with wheat. It's going to be a bit more tricky with a long-term crop like trees, but we really hope that we've made some good starts, some good inroads into getting there. So Lindsay's talked a little bit about Phytophthora pluvialis and Douglas fir, and one of the things that I keep hearing during this conference is that we try and look at these things in a 1-to-1 ratio, you know, Phytophthora pluvialis on pine, Phytophthora agathodicida on curry, but actually a lot of the time these things aren't working in isolation, and this is the case also with pluvialis and Douglas fir. So when Mireia was starting her PhD, she very quickly realised that the Swiss needlecast pathogen is always present, and it's something that she had to take into account when she was doing her experiments. So we're beginning to get this understanding that it's actually impossible to consider these systems in a one-to-one relationship. We actually need to start understanding the implications of these multiple organisms and the different interactions. And this has been going on throughout the life of this Healthy Trees, Healthy Future project. We've found this interaction between red needlecast and Swiss needlecast. With our genomics, we've had this consistent identification of different bacterial species associated with the different Phytophthora. Natalie's already referred to the association between physiological needle blight and Dothostroma needle blight out in the field, and what we see between the field and the lab. And this is something that's reoccurring in science in general. So we're starting to think about these things in a more holistic way. What are the different kinds of organisms that associate with these plants in the different parts of the plants? So this is all really exciting, but we need to think about what information might we be missing if we keep looking at these things in a one-to-one way, in a one-to-one interaction. So this is leading on to some new work, some new proposals that we have underway to look at these interactions in this more holistic way. So watch this space. I think it's going to be really exciting. So I briefly want to move on and just talk about some of the metabolomic analysis, just to touch on some results briefly. So this is work that's been done by Laura Raymond and Stefan Hill at Sion. So if we look at Cody initially, some work's been done with Cody tissue culture where the tissue culture was inoculated with these three different Phytophthora species, Phytophthora agathodicida, which causes Cody dieback, Phytophthora cinnamomi, and multivora, which are sometimes associated in those soils of Cody as well. Now what we see when we do this metabolomic analysis is that in the controls we don't see any induction of these particular chemicals, but we do see an induction of defence chemicals in the Phytophthora infected Cody. Interestingly, those defence chemicals are induced to a lower extent when the Cody tissue culture is infected with Phytophthora agathodicida. So maybe this is how agathodicida can be such an aggressive pathogen on Cody, is that it doesn't actually make Cody induce those chemicals that assist in defence. Now when we look at chemical analysis along an apple twig, this was done with some Braeburn twigs, and what was done was the twig was infected at one end, and then chemical analysis was done along the length of that twig. So the top line is the control, so there was no infection. The second line is uninfected tissue. So on the infected twig, the sample was taken a long way away on that twig from where the inoculation was done. The infection margin is where that the infected area and the non-infected area interact. And then the infected tissue is obviously the lesion, the yucky tissue where the infection has occurred. And you can see that there's no induction of chemicals in the control compared to the infected margin and the infected tissue. But then when we look at the uninfected tissue, there is an induction of chemicals there, and this is a long way from where the pathogen has been. So the plant is actually communicating through its cells, inducing chemicals before the pathogen even gets there. So this was a really interesting observation. Now when we look at the chemical analysis of pine, so we go back to the freezer with those samples and do this with, I think this was done with NMR, we see that we do get a difference in the types of chemicals that we see with the infected but compared to the uninfected samples. But unfortunately, we're not at the stage where the chemical databases can allow us to identify what those chemicals are. So this is still going to be a work in progress. We haven't quite got there with this one. All right, I'm going to hand back to Lindsay, who's going to go over the final achievements.